if you have synthesized any new drug from your chemistry lab, or if you have identified some uh, phytotherapeutic potential chemical, or if you have isolated some kind of drug from, am I audible you clearly? Audible, Aina? And please mute yourself because I used to get disturbed. Okay? Please mute yourself. Now suppose that you have identified any chemical structure or any drug from herbal origin, or if you have synthesized any new drug and you got a very good result in the preclinical studies. So आपने कोई ड्रग सिंथेसाइज किया या फिर कोई आइसोलेट किया, उसके बाद में आप उसका आपका एक्टिविटी है या नहीं वो चेक करते हो। अगर उसको एक्टिविटी है अच्छे से, सो व्हाट हैपन्स, आप जो है वो क्या करते हो फिर आप जो है वो प्री क्लिनिकल स्टडीज करते हो। सो यू अंडरगो प्री क्लिनिकल स्टडीज एंड यू यूज टू इवेल्युएट दैट ड्रग इन द एनिमल स्टडीज। ओके, कुछ एनिमल्स जैसे फ्रॉग हो गया, रैट हो गया, रैबिट हो गया, माइस हो गया, गिनी पिग हो गया, कोई मंकीज हो गए। सो यू यूज टू इवेल्युएट दैट ड्रग whether it is efficacy, it is having efficacy or not for a particular ailment, particular disease. Then you used to check in the pre-formulation, you used to check in the preclinical trials. Now what happens, if the drug is okay, if it is having very good result in the preclinical studies, then what happens, you think this drug should be formulated now. Now you think about the formulation. Now उसको वो ड्रग का फॉर्मुलेशन बनाना है क्योंकि आपको क्लिनिकल ट्रायल्स के लिए परमिशन ग्रांट हो चुका है, and for your clinical trials of your drug, so they has permitted the clinical trials, and now you have to use the drug in a various dosage form. For that particular purpose you have to take into take into consideration certain points regarding your drug molecule, regarding your excipient. So whatever the background work is done to make a effective formulation, effective formulation you have to do certain background work. आपको सब कुछ मालूम करना पड़ता है कि ये ड्रग का सोलिबिलिटी कितना है, ये ड्रग का पार्टिकल साइज कितना है, पार्टिकल साइज डिस्ट्रीब्यूशन कितना है। तो ऑल दीज पैरामीटर्स they you have to evaluate that parameters before commencing any formulation. Before commencing any formulation you have to do certain study regarding your drug molecule, regarding your active pharmaceutical ingredient, and regarding the excipient which are you are going to use in the formulation. So that study is nothing but the pre-formulation study. That study is nothing but the pre-formulation study. So after identifying a potential drug, see, there are different departments in the chemical, departments in the pharmaceutical engineering. Pharmaceutical industry में बहुत सारे डिपार्टमेंट होते हैं, जैसे R&D डिपार्टमेंट, रिसर्च एंड डेवलपमेंट डिपार्टमेंट, फॉर्मुलेशन डिपार्टमेंट, क्वालिटी कंट्रोल डिपार्टमेंट. व्हाट इज द फंक्शन ऑफ रिसर्च एंड डेवलपमेंट डिपार्टमेंट? R&D डिपार्टमेंट का क्या फंक्शन होता है? So they used to identify, they used to find out certain drugs, they used to synthesize the new drug, they used to isolate the new drug, they used to quantify it, they used to optimize that molecule for particular action by using SAR studies and etc. After development, after identification of new potential drug, then drug uh, they are tested for preclinical studies. And if you got a very good result in the preclinical studies, then you think about the clinical studies. Then you apply for clinical studies, there is a clinical trials, and for that you have to design the dosage form. Okay, so there are different kind of dosage form, you know that very well, I'm not going to repeat it. That is nothing but the oral dosage form, injectables. Okay, in the oral you have tablet, capsule, syrup, suspension, emulsion. Okay, then there are different kind of dosage form. So you have to formulate that drug in a different dosage form. So to formulate that drug, it is a function of your formulation development department. Now what they do, so they used to study certain properties of your drug. आपके ड्रग का प्रॉपर्टीज क्या है, मतलब उसका मोलिकुलर वेट कितना है, उसका पार्टिकल साइज डिस्ट्रीब्यूशन क्या है, ठीक है, उसका सोलिबिलिटी क्या है, उसका डिसोसिएशन कांस्टेंट क्या है, उसका मतलब पूरा पॉलीमॉर्फिक स्टडी होता है उसका, उसका क्रिस्टल शेप है कैसा है, इट इज whether it is amorphous, whether it is a crystalline. So they used to do this study because these parameters they affect the formulation. They affect the formulation. That's why you have to do this kind of study known as a pre-formulation. Now suppose that again this you can see in this diagram. It is a flow diagram for new drug development, and then how there is development of investigational new drug. So your industry has a different kind of department like medicinal and medicinal chemistry and pharmacology department, pre-formulation and research department, or formulation development department. We have pre-formulation research department, or then formulation development department, then process research and development, then analytical research development department, and toxicology and metabolism study department. So what happens? Medicinal chemistry and pharmacology department, they used to identify a particular certain potential chemical or certain potential molecule. So that is nothing but the drug discovery. That is the another part. There are a lot of different. You can take around 2 lectures on drug discovery. So there are different methods for drug discovery. There is another part. Now once there is a new drug, okay. Now what happened? So pre-formulation department, it is handed over to pre-formulation department. The formulation department does this kind of literature survey or data. They used to collect the information from various literature available. So literature search of preliminary data. So what is the molecular weight? What is the solubility? What is the key solubility data? So they used to identify this. And if they find that certain this अगर आपने कोई ड्रग जो आया है आपका तो वो अगर आपको मिला कि ये यार इसकी सोलिबिलिटी कम है so if that drug is having less solubility so what a formulation development department they used to again communicate with the research department. They used to again communicate with R&D and they used to tell so please increase the solubility of this molecule because if we formulate this molecule it is not going to be having good bioavailability. Again that molecule is handed over to R&D. So R&D what will they do? They will do molecular optimization. They will do molecular by different process like they will form a salt or they will form a solvates or they will make a crystalline compound into amorphous or they will formulate that they will cause esterification or prodrug and again that will be evaluated and again that will be requested for formulation level. Then now your your formulation development department what they do they used to take this molecule okay they used to study a different characteristic of that. ठीक है आपके यहाँ पे आपका एक नया ड्रग आया आपको ठीक है नया ड्रग मिला है आपको सब कुछ पैरामीटर्स ओके कि ये बायो अवेलेबिलिटी होता है इसका सोलिड डाटा अवेलेबल है molecular weight वगैरह preliminary डाटा होता है preliminary डाटा if it is available that is handed over to formulation department. Now pre-formulation department they used to study the physical properties of drug like bulk properties, solubility. Bulk property means particle size distribution, density, then porosity, etc. Then solubility profile, then stability profile. Stability profile in the sense that is degradation studies of that drug, oxidation, reduction, hydrolysis, photolysis. तो ये सब ड्रग आने के बाद प्री फॉर्मेशन डिपार्टमेंट आपका क्या करेगा सो दे विल स्टडी द फिजिकल कैरेक्टराइजेशन ऑफ दैट ड्रग देन after that will be if after that study is satisfactory if that study is satisfactory and it is meeting all the required standards then there will be formulation development and at that time also compatibility and stability is checked dissolution study is done bioavailability study is done then after that there is another phase first formulation and here the intrinsic dissolution stability study bioavailability study and scale up means you are initially doing only small batch then after that you are doing scale up in relatively large amount of tablets again there is a process research means so they will do one thing they will try to improve the yield they will try to find out the alternate route so if it is not satisfactory they will produce in a the product will be produced in the bulk then there is another process development or bulk scale up and another department is there known as the analytical research department so what they do they will take care of the method development of analysis so analytical method development like bulk clearance toxicology potency formulation assay IND formulation stability that is done by your analytical research formulation development क्या करेगा जो formulation development department है वो क्या करेगा जैसे ही R&D से ड्रग आता है आपका वो ड्रग का पूरा पैरामीटर चेक करेगा मतलब सोल्युबिलिटी ठीक है स्टेबिलिटी डाटा सोल्युबिलिटी डाटा लिटरेचर उसका जो भी लिटरेचर अवेलेबल है उसके ऊपर सर्च करेगा उसके बाद में अगर लगता है उनको कि ये ड्रग का थोड़ा सा मॉडिफाई करना जरूरी है क्योंकि इसका बायो अवेलेबिलिटी कम है तो वी हैव टू एनहांस द बायो अवेलेबिलिटी so that drug always again sent to a R&D department R&D department will do certain changes R&D department will do certain changes and after doing the changes and after getting a satisfactory result then again it is handed over to formulation pre-formulation research they will do the physical characterization like bulk properties solubility profile stability etc and formulation development will do their own accessibility or test then it will commence the formulation so normally this is the process ये नॉर्मल प्रोसेस होता है। तो इसके साथ साथ में प्रोसेस रिसर्च वाले भी काम करेंगे, एनालिटिकल रिसर्च वाले भी काम करेंगे इसके ऊपर, मेथड डेवलप करो, बायो अवेलेबिलिटी वाले भी, मतलब बायो फार्मास्यूटिक वाले भी काम करेंगे। देन ओनली देयर इज इन्वेस्टिगेशनल न्यू ड्रग। IND जैसे अपने अप्लाई किया, आपको फॉर्मुलेशन डेवलपमेंट करना। Now if you find a drug, if you find a drug with a promising effect in the animals, that promising effect in the animals means preclinical study that assures you or that give you warranty regarding the you can use this drug for the evaluation in the humans, कि आपको प्री क्लिनिकल स्टडीज में अगर अच्छा रिजल्ट आता है तो आप क्लिनिकल स्टडीज के लिए जाना चाहिए, मतलब क्लिनिकल ट्रायल्स के लिए। सो प्री फॉर्मुलेशन मेथड डील्स विद द फिजिको-केमिकल प्रॉपर्टीज ऑफ द ड्रग। What is pre-formulation? It deals with the physico-chemical properties of the drug that could affect the performance of your performance and development of in the patient dosage form. So whatever dosage form you are designing, so this pre-formulation studies deals with the different physico-chemical properties of drug which can affect the formulation. Now you have to understand these properties. Understanding these properties of pre-formulation, they helps in the designing of a better dosage form. They helps in the designing and support of molecular modification if any required. After that, pre-formulation is done. After that, pre-formulation is done. Study what kind of study is there? I will explain. Pre-formulation is done for what is the main aim of pre-formulation? So pre-formulation is done for these three reasons. First, to know the physical and chemical properties of newly developed drug or newly synthesized drug. To know the physical and chemical properties of your newly identified drug, then to know the kinetics and stability properties of that drug, to ensure the compatibility and compatibility with the common excipient. So you are designing a dosage form, you are making a new tablet. So for making a tablet, you need an excipient. For making a syrup, you need an excipient. For making a different kind of dosage form, you need an excipient or additives. So you have to ensure that your drug is compatible or your common excipient or not. ठीक है, तो आपको ये सब फिजिकल केमिकल प्रॉपर्टीज उसका स्टडी करना पड़ेगा प्री फॉर्मुलेशन में, टू नो द फिजिकल केमिकल प्रॉपर्टीज ऑफ द ड्रग, टू नो द काइनेटिक्स एंड स्टेबिलिटी, वो ड्रग कैसे एब्सॉर्ब होता है, कितना एब्सॉर्ब होता है, स्टेबिलिटी कितना है उसका, वो चेक करना पड़ता है प्री फॉर्मुलेशन। टू इंश्योर द कंपेटेबिलिटी विद द कॉमन एक्सपेंट, आप जब फॉर्मुलेशन बनाएंगे, एक्चुअल फॉर्मुलेशन बनाएंगे, तब जो आप एक्सपेंट यूज करने वाले, जैसे टाल्क यूज करते हो, ग्लाइडेंट यूज करते हो, डिसइंटीग्रेंट यूज करते हो, बाइंडिंग एजेंट यूज करते हो, so whether those additives are compatible with your drug or not, that is checked in the pre-formulation. So pre-formulation is done for three reasons: to know the physical properties of new drug, to know the kinetics and stability, to ensure the compatibility of your drug with the commonly used excipient. Now pre-formulation, because this pre-formulation has a very much, it has a much impact on manufacturing, then storage and performance of your dosage form or drug product. So pre-formulation has an impact on the manufacturing, then storage performance of the dosage form. Now pre-formulation provides a tool. Okay, so if you have done pre-formulation studies, so pre-formulation provides a way. It provides a way by which drug should be processed and stored. Okay, to ensure the quality of that product. Okay, so pre-formulation क्या करता है? Pre-formulation study ensures you that it ensures you or it will provide a way by which a drug product should be processed. आपको एक टैबलेट बनाना है, किस तरीके से बनाना है आपको, उसका स्टोरेज कैसे होना चाहिए, to ensure the quality of that product. So pre-formulation provides a way by which product should be processed and stored and to ensure the quality of that drug. So what are the different studies done under the pre-formulation? So primary findings and molecular optimization. जैसे मैंने बोला आपको R&D ने कोई ड्रग बनाया तो वो डायरेक्टली प्री फॉर्मुलेशन डिपार्टमेंट को जाता है, फॉर्मुलेशन डेवलपमेंट डिपार्टमेंट, तो वो चेक करते हैं उसका फिजिकल इंपोर्टेंट प्रिलिमिनरी पैरामीटर्स, उसमें अगर कोई लगता है कि कुछ मॉडिफिकेशन करना है तो वो फिर से बाद में R&D को हैंड ओवर करते हैं. So primary findings are molecular optimization. Primary findings, okay, finding of known properties, dosage form, therapeutic area, potential, relative, competitive. So you have to find in the literature, okay, what is the available property. To know the finding of known properties, different dosage form of that particular drug, therapeutic area, okay, मतलब किस में यूज करना है, potential relative to the competitive product. कि ऐसा कोई कॉम्पिटिटर है क्या कि सेम ड्रग का आपने तो ओके सो केमिस्ट्री डिपार्टमेंट should find out the molecular weakness of that molecule and optimize the compound. केमिस्ट्री डिपार्टमेंट का काम क्या होता है कि R&D में कि उसका काम है कि नया मॉलिक्यूल बनाना या और बनने के बाद वो मॉलिक्यूल का वीकनेस क्या है whether it is easily hydrolyzed in the external environment or acid environment or basic environment. So they have to optimize that compound. They have to make certain changes in that compound to make the compound stable. They have to study probable degradation mechanism. They have to study probable decay mechanism and condition for degradation. तो मतलब ये ड्रग जैसे स्टमक में जाएगा ये टिकेगा या नहीं कि वहाँ पे जाके डिग्रेड होगा इंटेस्टाइन में गया तो वहाँ पे भी डिग्रेड होगा क्या ऐसा पूरा चेक करना पड़ता है so it is useful for stability test okay they do stability test method for stabilization so if compound is unstable they will modify they will okay they will modify and they will stabilize the compound okay for example aspirin जो है it is nothing but the acetyl salicylic acid so it is a ester so ideally used to it will undergo hydrolysis optimize that compound that is the function of your chemistry department. Again, this formulation is nothing but a confirmation that there is no significant hurdle for formulation. Then first we have to study the solid state physical properties of drug. Whatever newly developed drug is there or whatever newly identified drug is there. So you have to study the solid state physical properties of that drug. Now in that you have different properties like compaction properties. कि वो ड्रग का मॉलिक्यूलर वॉल्यूम कितना है डेंसिटी कैसा है रिफ्रैक्टिव इंडेक्स है कितना है thermal and electrical conductivity hygroscopy so these property molar volume density refractive index thermal and electrical conductivity hygroscopicity so it comes under the compaction properties then again thermodynamic properties like melting point sublimation melting and sublimation temperature internal energy of the compound enthalpy heat capacity entropy free energy chemical potential thermodynamic activity vapor pressure solubility so these properties comes under the thermodynamic properties. Then spectroscopic evaluation of that drug, whether okay, so UV absorption, then IR absorption, then microwave region में rotational spectra in the microwave region, then nuclear spin resonance that is NMR transitions. So you have to deal with this so solid state physical properties, compaction properties, so molecular volume, density, refractive index, okay then thermal and electrical conductivity, hygroscopicity of that material. Thermodynamic property like melting point sublimation, melting temperature, and heat capacity of that compound, enthalpy, entropy of that compound, free energy, chemical potential, thermodynamic activity, vapor pressure, and solubility. So these are thermodynamic properties, and you have to also study the spectroscopic properties, whether that drug is having particular lambda max or not. कि वो if drug is having whether that drug is active in the UV or not. If that is active in the UV, that means it has a conjugation. If that is not active in UV, that means it does not have conjugation. It absorb certain radiation in the IR. Then what is the pattern? What is the NMR spectrum of that compound? That is the spectroscopic analytical techniques or properties. Then kinetic properties you also study like what is dissolution rate, solid state reaction, uh, solid state reaction rate, then stability, etc. So those are the kinetic properties. Now surface properties like what is surface force, surface interfacial tension, what is crystallinity. So you have to study this property okay, in the solid state. Mechanical properties like hardness, tensile strength, compactability, okay, compatibility, then flow properties and blending of that active drug. Okay. So you have to deal with these physical state properties, solid state physical properties. Now salt formation. Now what is now from this point onward so they can ask you question in the GPAT. Already they have asked the questions. So ये इसके आगे सब जीपेट के लिए इम्पोर्टेंट है ये पहला का तो जीपेट के लिए इम्पोर्टेंट नहीं था ये जस्ट आपके पर्पस के लिए था आपको थोड़ा सा एक स्टार्टअप होने के लिए नाउ दिस salt formation means making a salt with the acid or base of your drug so whether your drug is basic or acidic that depends organic compound by abstracting the proton organic compound they used to abstract the proton from ionized drug and they used to neutralize it with the counter ion. For example, ephedrine hydrochloride. So ephedrine क्या है? Ephedrine is alkaloid. It has a secondary nitrogen. So withdraw that proton from that nitrogen and neutralize that reactive species with the HCl. So epidine hydrochloride, that will be a salt. Okay, because you know that salt form of drug will have good water solubility. जैसे salt form होता है उसका water solubility क्या होता है, ज़्यादा होता है. Generally organic salts are more water soluble than unionized drug, which offers higher dissolution rate and bioavailability. So generally salt form of drug is water soluble as compared than ionized drug, and it will give you better dissolution and better bioavailability. Okay, so common anions like negatively charged ions like hydrochloride, bromide, chloride, iodide, citrate, malate, phosphate, sulfide, then pampate, etc. They are used for the salt formation. आपने देखा होगा इपिड्रीन hydrochloride, morphine sulfate, ferrous fumarate. So fumaric acid, that is the anion, and it is formed salt with the ferrous. So that is salt formation. Then again, cations like magnesium, calcium, sodium, zinc. So they are nothing but the cations used in the salt formation. So these are the anions used in the salt formation. That is the hydrochloride, then bromide, chloride, iodide, citrate, sulfate, phosphate, etc. Okay. So salt form of drug is more water soluble. Now pre-formulation is divided into these three study. So in the pre-formulation study, what kind of study you have to do? So these three. आपको pre-formulation में ये पढ़ना है क्या? Solubility studies, you have to do solubility studies. So it is done by doing the study under. You have to determine the pK, dissociation constant. आपके कंपाउंड का डिसोसिएशन कांस्टेंट कितना है, कैसा है वो. सो pKa determination, then pH solubility profile, then common ion effect, etc. In that also common ion effect of temperature on solubility of drug, then solubilization mechanism if drug is insoluble in water, so you have to improve the solubility. For that solubilization mechanism are there. Then what is the partition coefficient of your drug, then what is the dissolution profile of your drug. So it comes under the solubility studies. Okay, so it comes under the solubility studies. Now bulk characterization. So in that about that physical state of drug. So crystallinity and polymorphism, crystallinity and polymorphism, then hygroscopicity of that drug, then micrometrical properties like particle size distribution, particle size characterization, etc. Density, porosity, etc. So density, porosity, then flow properties of powder, so all this comes under the bulk characterization. Lastly, you have to study the stability of that drug. Okay, stability of drug. So solution stability or solid state stability or drug excipient compatibility. So your pre-formulation chapter deals with this study. Again you see यहाँ पे एक बार और भी देख लो कि पहला क्या है. ये तीन तीन टाइप की स्टडी आपको करना है प्री फॉर्मुलेशन में. सोलिबिलिटी स्टडी, तो उसमें आपको पीके डिटर्मिनेशन करना है, पीके मेजर करना है. पीएच सोलिबिलिटी प्रोफाइल क्या है आपका ड्रग का, कि पीएच इंक्रीज होगा तो सोलिबिलिटी क्या होगा. देन कॉमन आयन इफेक्ट, ओके, पीएच सोलिबिलिटी प्रोफाइल एंड कॉमन आयन इफेक्ट, effect of temperature, solubilization, then partition coefficient, dissolution. In the bulk characterization, you have crystallinity and polymorphism, hygroscopicity, then micrometric properties like particle size characterization, density, porosity, powder flow properties. And in the stability analysis, you have solution stability, solid state stability, and drug excipient compatibility. So first is nothing but the bulk characterization. In that you have crystallinity and polymorphism. Crystallinity and polymorphism. So we have already seen one chapter in the pharmaceutical engineering. If anybody has not seen that, they can go and see that chapter. Known as a, we have seen that crystallization chapter. So in that we have again we have already explained these things, but I am going to again explain it. So polymorphism. What is meant by polymorphism? It is the property of compound to undergo different crystallization, different crystal. So polymorphism is nothing but the property of compound to crystallize more than one crystal form. So property of compound to crystallize in more than one different crystalline form, that is a polymorphism. So compound, whatever is your drug or new drug, okay. So compound under microscope, you have to see it under microscope. Now after seeing in the microscope, first that compound will be either transparent, कि वो transparent रहेगा, either it will be transparent or it will be non-transparent. So okay, it will be non-transparent. कि आप माइक्रोस्कोप के नीचे आप कंपाउंड को देखोगे तो वो तो ट्रांसपेरेंट होगा या फिर नॉन ट्रांसपेरेंट. Now if it is transparent, then they are known as a bifranges. So in that also that again if it is transparent, it may be either anisotropic or isotropic. So anisotropic matlab kya? So if your compound is having one refractive index, so if your compound is having one refractive index, known as the anisotropic. Okay. Now if your compound has a okay, if your compound has a only one refractive index, it is known as a isotropic. And if your compound has more than one refractive index, it may be either 1, 2, 3, 4, etc. So okay, that is anisotropic. Anisotropic means a compound having more than one refractive index. Isotropic means compound having only one refractive index. So if your compound has anisotropy, that means having more refractive index. So again they are further divided into two. So if your compound has only two refractive index, it is known as a uniaxial. And if your compound has a three refractive index, it is known as a biaxial. Now, it is a ट्रांसपेरेंट कंपाउंड. अगर आपका कंपाउंड ट्रांसपेरेंट है माइक्रोस्कोप के नीचे, तो उसको क्या बोलेंगे? बायफ्रिंजेस. बायफ्रिंजेस, ठीक है. और उसमें भी दो टाइप का आता है. बायफ्रिंजेस मतलब उसको रिफ्रैक्टिव इंडेक्स है. तो अगर जो एक से ज्यादा रिफ्रैक्टिव इंडेक्स है तो उसको बोलेंगे अनिसोट्रोपिक. अगर एक ही रिफ्रैक्टिव इंडेक्स है उसको बोलेंगे आइसोट्रोपिक. तो अनिसोट्रोपिक अगर है तो उसमें दो टाइप का आता है. यूनिएक्सियल मतलब कंपाउंड को दो रिफ्रैक्टिव इंडेक्स है. बायएक्सियल मतलब तीन रिफ्रैक्टिव इंडेक्स. नाउ सेकंड इज नथिंग बट इफ योर कंपाउंड इज नॉन ट्रांसपेरेंट, आपका कंपाउंड नॉन ट्रांसपेरेंट है अंडर माइक्रोस्कोप. नाउ इट इज अगेन डिवाइडेड इनटू टू स्ट्रक्चर. टू ओके. सो एक्सटर्नल स्ट्रक्चर known as a habit. External structure of their compound is known as a habit, and internal structure. Internal structure. External structure is known as a habit and internal structure. इसके ऊपर क्वेश्चन आ चुका है. External structure of compound is known as a habit. So you have to remember this. Internal structure is divided into two subtypes again. So internal structure may be either crystalline solid or it may be either amorphous. So crystalline means what? Crystalline solid or crystal means what? Crystalline form of your compound means the particles of that compound is arranged in the regular geometric fashion, or there is a symmetric particle arrangement in the crystal lattice. So that is known as crystalline. It has a very low energy because यहाँ पे crystalline में क्या होता है कि वहाँ पे सिस्टमिक अरेंजमेंट होता है पार्टिकल का, तो उसमें एनर्जी भी कम रहता है. एनर्जी कम हो गया है तो उसका सोलिबिलिटी भी क्या होता है? कम होता है. तो इसके ऊपर भी क्वेश्चन आ चुका है. वन ऑफ द फॉलोइंग फॉर्म ऑफ कंपाउंड इज मोर सोलिबल. तो याद रखो यहाँ पे क्रिस्टलाइन फॉर्म जो है दैट इज अ सिस्टमिक अरेंजमेंट, दैट इज हैविंग सिस्टमिक अरेंजमेंट ऑफ particles in the crystal lattice. It has a very low energy and that's why it has a low solidity. Another form of internal structure is nothing but the amorphous. Okay, amorphous means what? So amorphous means, so this form is having random arrangement of particles in the crystal lattice, and that's why it has a very high energy and it has a very high solubility. तो यहाँ पे देखो amorphous का solubility ज़्यादा रहता है, crystalline का कम रहता है, क्योंकि crystalline में क्या है? सिस्टमिक अरेंजमेंट रहता है पार्टिकल का और उसका एनर्जी लो रहता है. लो एनर्जी मतलब लो सॉलिबिलिटी. यहाँ पे एनर्जी हाई रहता है. यहाँ पे रैंडम अरेंजमेंट होता है और एनर्जी क्या रहता है? हाई रहता है. दैट्स व्हाई हाई सॉलिबिलिटी. सो amorphous compounds, they are prepared by different methods like rapid precipitation, lyophilization, and this rapid liquid cooling. So it is the method for preparation of your amorphous compound. So amorphous form of drug is prepared by this method. Now you may ask, आप पूछेंगे सर amorphous form क्यों बनाना है? ठीक है. amorphous form क्यों बनाना है? Because amorphous has a more solubility, crystalline has a less solubility. So that's why to enhance the solubility or to increase the solubility of poorly soluble compound and to enhance the bioavailability. So sometimes you have to make the you have to use the amorphous form of your drug. Okay, so that's why you should know this method: rapid precipitation, lyophilization, and rapid cooling. ये बहुत ही important है. नाइपर में तो बहुत बार क्वेश्चन आ चुका है. Then crystalline. So crystalline is a systemic arrangement. It will have low energy, low solidity, and again it is divided into two: single polymer. It may be either single polymer. It may be either single polymer. It may be either molecular adduct. Single polymer or molecular adduct. Molecular adduct means what? Molecular adduct means it may be either single polymer or molecular adduct. So we'll see first single polymer. Single polymer मतलब एक ही polymer पे उसका crystalline structure है और एक ही polymer पे. So इसमें भी दो टाइप आता है. So in the single polymer it may be either monotropic or anisotropic. It may be either monotropic or anisotropic. So what is meant by monotropic? All polymorphs, they are they are unstable at a temperature and pressure. If okay, so all polymorph of this monotropic compound is unstable. Okay, and all in isotopy means all polymorphs are isolable and they are stable. Single polymorph again to monotropic and anisotropy. Monotropic means their polymorphs are unstable, and anisotropy means all polymorphs are isolable. Okay. Then if there is a, okay, it was a single polymorph. Now molecular adduct. So they comes under again two. So if while crystallization there is entrapment of solvent, crystallization process में क्या होता है कि कभी-कभी उसमें उसमें क्या होता है कि कभी-कभी सॉल्वेंट का एंट्रैपमेंट होता है ड्यूरिंग द क्रिस्टलाइजेशन. क्रिस्टल के बीच में कुछ सॉल्वेंट का मॉलिक्यूल्स जाता है, ठीक है. सो दैट्स व्हाई दे आर मोलिकुलर अडक्ट. अडक्ट मतलब प्लस, अडक्ट मतलब एडिशन, ठीक है. तो मॉलिकुलर अडक्ट मतलब क्या, जब मॉलिक्यूल बनता है कम क्रिस्टलाइज होके उस टाइम पे जब सॉल्वेंट उसमें ट्रैप हो जाता है, दैट इज नोन एज मॉलिकुलर अडक्ट. Now they are further divided into two types, non-stoichiometric and stoichiometric. So non-stoichiometric means what? Non-stoichiometric means, so if for example non-stoichiometric means okay, stoichiometric means 1:1. Okay, stoichiometric means 1:1. अगर आपका एक मोल है आपका कंपाउंड और उसके साथ में एक मोल वाटर का है या फिर एक मोल जो भी सॉल्वेंट है, तो 1:1 एडिशन इज नोन एज stoichiometric. And 1:1, if it is not a 1:1 then it is known as a non-stoichiometric. यहाँ पे सॉल्वेंट और सोल्यूट अगर सेम क्वांटिटी में ऐड हो चुके हैं तो उसको बोलते हैं स्टोइचियोमेट्रिक. अगर नहीं है तो उसको नॉन स्टोइचियोमेट्रिक. सो 1:1 combination is known as a stoichiometric. So it is again further solvents and hydrate. Now solvents means what? Solvents means, so if the solvent used for crystallization that traps into the your crystal, that is known as solvents. And if that solvent is nothing but the water, अगर वो solvent water है तो उसको बोलेंगे hydrate. तो stoichiometric means 1:1. okay, जैसे मान लीजिए कि एक पार्टिकल है आपका और एक वाटर का मॉलिक्यूल है. तो if that one particle and one water molecule forms a crystal, that is known as a hydrate क्योंकि वो water है. If solvent is a benzene or anything else, okay, so anything else, so if that solvent one molecule and particle one molecule forms a crystal, that is known as a solvents. So solvents means solvent, it is for solvent, and hydrate means it is for a water molecule. So then again non-stoichiometric means non 1:1 combination. So in that you have cage, layer and channel. Layer and channel इसका arrangement होता है cage, layer and channel. So this is the polymorphism. ये समझ में आया आपको? Then again next one is a, you can see examples of crystal habit over here. Habit means what? Habit means external. Habit means external structure of your crystal. So there are different kind of external structure like acicular, angular, bladed, crystalline, dendrites, fibrous. Okay. So acicular means elongated prism. तो एक prism के जैसे लंबा like a needle-like structure. Needle-like crystal, they are known as acicular crystal. Angular means what? Sharp-edged, roughly polyhedral structure known as angular. Bladed means flattened, acicular. Crystalline. Actually, these are the description of shapes of your crystal. Crystal के shapes कैसे होते हैं उसका description है ये. So not much important. Again, crystalline, fully developed geometric shape. Crystalline means what? Fully developed geometric shape in the. Dendrites means branch crystalline. Fibrous means regular or irregular thread-like structure. Then flaky or platy means flakes or plates. Plate and slat-like. Granular means what? Granular means equidimensional irregular shape, that is a glandular. Then irregular means lacking any symmetry. कोई अगर सिमेट्री नहीं है तो उसको बोलेंगे irregular crystal. Nodular means rounded irregular shape. Prismatic मतलब geometric prism. Then spherical मतलब global shape रहेगा उसका. So these are just examples of some habit. Okay, कि external shape कैसा रहेगा. So already we are seeing in the crystallization chapter. Now what is polymorphism? Polymorphism is nothing but the ability of compound to crystallize more than one crystallized form. तो अगर आपका कोई कंपाउंड है उसके दो तीन टाइप के क्रिस्टल बनते हैं क्रिस्टलाइजेशन के, वो तो उसको बोलेंगे पॉलीमॉर्फिज्म. सो एबिलिटी ऑफ कंपाउंड टू क्रिस्टलाइज मोर देन वन क्रिस्टलाइन फॉर्म. सो दिस crystals you see these crystalline form or polymorph. Polymorph one crystalline form is a polymorph 1, second crystalline form is a polymorph 2, third one is third. So these polymorphs they differs in the physical chemical properties again. कि हर एक polymorph का जैसे जैसे मान लीजिए कि barbiturates है. Barbiturates these are the drugs which shows 90% polymorphism. उनमें 90% polymorphism आता है. तो वो डिफरेंट क्रिस्टलाइन फॉर्म में क्रिस्टलाइज होते हैं. तो हर एक क्रिस्टल का अलग अलग फिजिकल प्रॉपर्टी होता है कि कोई क्रिस्टलाइन शेप, कोई क्रिस्टल का सोलिबिलिटी ज़्यादा होगा, किसी का कम होगा. किसी का मेल्टिंग पॉइंट कम होगा, किसी का ज़्यादा होगा. So these polymorphs they differs in their physicochemical properties, and that's why they can affect the bioavailability. That's why you have to study polymorphism. For example, chloramphenicol palmitate. Chloramphenicol palmitate is a ester of chloramphenicol with palmitic acid, and it has a three crystalline form A, B and C. क्लोरमफेनिकॉल palmitate नाम का जो ड्रग है, क्लोरमफेनिकॉल का ईस्टर है palmitic acid. उसका क्या है, तीन पॉलीमर्फ्स है A, B, C एंड अमorphous फॉर्म. मतलब ये क्रिस्टलाइन फॉर्म उसका तीन आता है एंड एक अमorphous फॉर्म. ओके, सो this chloramphenicol palmitate has three crystalline form A, B, C and amorphous form. So out of that, your crystalline form B is a stable one. You have to isolate this stable crystal and then use that crystal in your formulation so that your formulation will be stable. अगर आपने फॉर्म A यूज किया क्लोरम्फेनिकॉल palmitate का, तो आपका फॉर्मुलेशन स्टेबल नहीं रहेगा, अनस्टेबल. C यूज किया तो स्टेबल नहीं रहेगा. So you have to use because B form is a stable. So there are other drugs, and if you have seen the or if you have studied the suppository, अगर आपको suppository के बारे में मालूम होगा, तो whatever the bases used in suppository, जो suppository base होते हैं, suppository base में, so whatever suppository base is there, so in that you have cocoa butter. Okay, in the suppository you have cocoa butter as a suppository base. So cocoa butter also, if you heat cocoa butter more than 35 degrees Celsius or 40 degrees Celsius, so it again will crystallize in a different crystallization, will show polymorphism. Okay, so it will crystallize in alpha, beta, gamma, delta, four crystalline form. Out of that, beta form is stable. Physical chemical properties of a drug with, okay, physical chemical properties of drug, they will vary, they will differ with the internal structure of the solid. Example, melting point, hardness, solidity, optical properties, vapor pressure. मतलब क्या कि जो polymorphism है, it is the ability of compound to form a different crystal, to crystallize in a different crystalline form. तो वो different crystalline form जो है उसको बोलेंगे polymorph. And these polymorphs they differs in the physical properties. उनका physical properties अलग अलग होता है. So in turn they will affect the your bioavailability, your product. Okay? So physical chemical properties of drug, they vary with the internal structure of solid. Example, melting point, hardness, solubility, optical properties. So these polymers will have a different property like melting point. दो पॉलीमर का अलग होगा. जैसे मान लीजिए कि आपका कोका बटर है, कोका बटर सपोजिटरी. So out of the, in that beta form is stable. So beta form, beta crystalline form है कोका बटर का, तो that will melt on body temperature, that is 37 degrees Celsius. If alpha form is there, if alpha form अगर generate होगा crystallize में, तो alpha form will melt at 20 degrees Celsius only. So that suppository will not be able to hold in the rectum. It will easily flush out. So that's why you have to give attention with the this particular polymer. कौन सा polymer stable है, कौन सा नहीं है. Now there are different methods by using different methods. By using different methods, you have to study this polymer, this pre-formulation. Pre-formulation के लिए आपको बहुत सारे टेक्निक आते हैं उसके लिए. So methods for quantitative analysis of physico-chemical properties of drug. कौन से मेथड यूज कर सकते हो, उसमें कौन सा प्रॉपर्टी आप मेजर कर सकते हो. सो फर्स्ट वन इज अ माइक्रोस्कोपी. सो इन माइक्रोस्कोपी you can observe the internal and external structure of your compound. You can okay, so you can also see other properties. So whatever sample is required for, there is 1 milligram sample is enough for doing a microscopic study. So fusion method is there, fusion method. So in this method you can determine the degradation temperature, okay, then melting temperature, etc. using fusion method. It is also known as a hot stage microscopy. Again यहाँ पे भी आपको 1 milligram sample enough है. DSC and DTA, differential scanning calorimetry and differential thermal analysis. Then you can see here the melting temperature, evaporation temperature, then boiling point, melting point using this DSC and DTA. So again 2 to 5 milligram of sample is required. Then in a higher scope spectroscopy, it will give you idea about the number of important functional groups. Okay, so sample required is a 2 to 20 milligram. Again this X-ray diffraction. So X-ray diffraction will give you idea about the crystal shape. So 500 milligram sample is required for X-ray. Scanning electron microscopy. Again whatever you can observe by microscopy, with the more preciseness you can observe by scanning electron microscopy. 2 mg sample is required. Thermogrammetric analysis, again 10 milligram sample is required. Solubility and dissolution analysis, sample from milligram to gram can be required. There is just a what are the different methods you utilize in the studying of physicochemical properties of drug. तो आपको ड्रग का फिजिको-केमिकल प्रॉपर्टीज जैसे फिजिकल प्रॉपर्टीज केमिकल प्रॉपर्टीज स्टडी करना है तो कौन से मेथड्स है माइक्रोस्कोपी है फ्यूजन मेथड है डिफरेंशियल स्कैनिंग कैलोरीमेट्री है डिफरेंशियल थर्मल एनालिसिस आईआर स्पेक्ट्रोस्कोपी एक्स-रे डिफ्रैक्शन स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोपी thermogravmetric analysis and mercury dissolution. You can see over again here methods and application for quantitative analysis of physicochemical properties of drug. So using microscopy you can determine the nature of substance. Then crystal morphology can be seen using microscopy. So again, okay. Then using fusion method or hot stage microscopy, you can study the crystal melting temperature, etc., etc. Then again from DSC, differential scanning calorimetry and the differential thermal analysis, you can study the polymorphism, crystal purity. Crystal purity is इसके ऊपर नाइपर में क्वेश्चन आता है. I will explain this. Crystal purity. So crystal purity is analyzed or determined by differential scanning calorimetry. क्रिस्टल कितना प्योर है कि उसमें इम्प्योरिटी है या नहीं. क्रिस्टल प्योरिटी इज मेजर्ड बाय यूजिंग डिफरेंशियल स्कैनिंग कैलोरीमेट्री. ओके, तो इसमें जब पीक्स आता है पीक्स ऐसे पीक आएगा वहाँ तो मोर विल बी द शार्प पीक मोर विल बी द प्योर क्रिस्टल. तो शार्प पीक इंडिकेट्स द प्योर क्रिस्टल. इसके ऊपर क्वेश्चन आता है. Then again from using IR spectroscopy you can measure the water content of the crystal, you can study the crystal, you can identify the groups, etc. By using X-ray diffraction, so you can able to see the crystal shape in outer surface of the crystal, etc. By using scanning electron microscopy you can study the crystal morphology. By using thermogravmetric analysis, again it is also utilized for study of crystal. By using NMR, you can study the crystal. By HPLC, you can analyze the stability. So HPLC analysis sometimes known as a stability indicating assay. इसके ऊपर पे नाइपर में क्वेश्चन आता है. HPLC is known as a stability indicating assay. Stability indicating assay. So stability after all you have to measure using this HPLC. Okay. Then dissolution and stability studies they are useful in the studying of bioavailability. These are the different methods and applications. Okay, these are the methods, their application in quantitative analysis of physicochemical properties. एक आपको ये थोड़ा सा मेथड याद रखना है. नाउ मॉइस्चर कंटेंट कि आपके ड्रग का मॉइस्चर कंटेंट कितना है, तो इसके ऊपर भी क्वेश्चन आता है 100%. सो मॉइस्चर कंटेंट मीन्स व्हाट? It is the amount of moisture that is held by or that is held by your compound. So amount of moisture that is held by your compound known as a moisture content. Now it may be either the compound may held moisture through its internal structure or by due to its external bond formation. So depending on that amount of moisture present in the compound, depending on that they are classified into two types: hygroscopic compounds and deliquescent compounds. What is meant by hygroscopicity? Hygroscopic compound means substance that can absorb the moisture and will that can absorb the moisture from air. ठीक है, अगर आप कंपाउंड आपका मॉइस्चर एब्सॉर्ब करता है एयर में से, तो इफ योर कंपाउंड एब्सॉर्ब द मॉइस्चर फ्रॉम एयर इट इज नोन एज हाइग्रोस्कोपिक. फॉर एग्जांपल आयोडीन वगैरह, जो आयोडीन का व्हाट इज मीन बाय डेलिक्वेसेंट? सो अगेन द कंपाउंड दैट हैज एबिलिटी टू एब्सॉर्ब द मॉइस्चर फ्रॉम एनवायरनमेंट बट टू ए सफिशिएंट एक्सटेंट सो दैट इट कैन गेट डिसॉल्व. मतलब क्या कि हाइग्रोस्कोपिक सब्सटेंस क्या करेगा, वाटर को एब्सॉर्ब करेगा मॉइस्चर में और उसका वेट इंक्रीज होगा. ठीक है, उसका वेट क्या होगा, इंक्रीज होगा. डेलिक्वेसेंट सब्सटेंस क्या है कि वो इतना ज्यादा मॉइस्चर एब्सॉर्ब करेगा कि वो पूरा लिक्विड बन जाएगा. सो हाइग्रोस्कोपी में सब्सटेंस दैट एब्सॉर्ब द मॉइस्चर फ्रॉम एयर और एनवायरनमेंट एंड इंक्रीजेस द वेट. एंड डेलिक्वेसेंट मींस दैट कैन सफिशिएंटली एब्सॉर्ब द मॉइस्चर टू कन्वर्ट सॉलिड इनटू लिक्विड. सो दैट इज अ डेलिक्वेसेंट. Change in the moisture, okay, changes in the moisture level significantly affects the chemical stability because presence of moisture in the compound may cause hydrolysis and all that. Okay, changes in the moisture level significantly affect the chemical stability. Then flowability, if there is more moisture in the granules, more moisture in the powder, it will not a free flowing powder. Then compactability, compression force भी उसको क्या लगेगा, ज़्यादा लगेगा. So changes in the moisture level significantly affect the chemical stability, flowability and compactability. Now इसके ऊपर GPAT में क्वेश्चन आ चुका है ऑलरेडी. As per the European Pharmacopoeial Technical Guide, as per the European Pharmacopoeial Technical Guide, so they have classified the degree of hygroscopicity. आपका कंपाउंड कितना हाइग्रोस्कोपिक है उसके ऊपर उन्होंने they have determined or they have classified the upon degree of hygroscopy the compound into 4 classes based upon the statistical method. What they do, so उन्होंने क्या किया कि कंपाउंड को 4 कैटेगरी में डिवाइड किया है, hygroscopic, very hygroscopic, slightly hygroscopic, deliquescent. As the European Pharmacopoeial Technical Guide, they has classified the degree of hygroscopicity into 4 classes based upon the static method after storage at 25 degrees Celsius for 24 hour at 80% relative humidity. तो क्या करते हैं कि आपका कंपाउंड जो है वो इस स्टोरेज कंडीशन में स्टोर करते हैं कौन से 25 डिग्री सेल्सियस 80% रिलेटिव ह्यूमिडिटी फॉर 24 आवर्स. ठीक है, प्री वेट कंपाउंड आपको कंपाउंड को वेट करेंगे और वेट करके नोट डाउन करेंगे, वो कंपाउंड वहां पे रख देंगे वो कंडीशन पे 25 डिग्री सेल्सियस 24 आवर्स एट 80% रिलेटिव ह्यूमिडिटी. उसके बाद में 24 आवर्स के बाद में फिर वो कंपाउंड वहां से लेके आएंगे और उसका वेट करेंगे. ठीक है, मतलब वेट मेजर करेंगे उसका बैलेंस को. पहले प्री वे क्या है, मतलब मान लीजिए कि पहले आपके कंपाउंड ने उन्होंने 1 ग्राम कंपाउंड लिया आपका वेट करके और बाद में 24 आवर्स को ये कंडीशन पे रखने के बाद में फिर से वेट किया, तो अगर उसका वेट 1.2 ग्राम हो गया मीट 200 मिलीग्राम उसने क्या किया है वाटर एब्सॉर्ब किया है एनवायरनमेंट से. सो डिपेंडिंग ऑन दैट दे हैव डिवाइडेड द कंपाउंड्स इनटू डिफरेंट कैटेगरी. सो दे हैव डिवाइडेड कंपाउंड इनटू first one is slightly hygroscopic. What do you mean by slightly hygroscopic? Now if your compound absorb the moisture and the weight is increased which is less than 0.2%. अगर आपने के कंपाउंड ने वाटर एब्सॉर्ब किया और उसका वेट इंक्रीज हो गया कितना, so depending on the European technical guide they have classified the degree of hygroscopicity in 4 classes: slightly hygroscopic, hygroscopic, very hygroscopic and deliquescent. Okay, so they have classified these 4 types by storing the compound at a particular condition known as a temperature is 25 degrees Celsius, humidity is 80% and they are stored for 24 hours. Now depending on that increase in weight is measured. So if increase in weight of that compound is less than 0.2% weight by weight, okay, but okay, so if increase in weight of that compound is more than 0.2% weight by weight but less than 2%. ठीक हो क्या यहाँ पे समझ में आना चाहिए आपको कि क्वेश्चन इसके ऊपर आता है, ऑलरेडी आ चुका है. मान लीजिए कि आपने वो कंपाउंड उस कंडीशन पे रखा है. ठीक है, कंडीशन पे रखने के बाद क्या बाद में नेक्स्ट डे आपको क्या करना है वेट मेजर करना है. Now if increase in the weight of that compound is more than this 0.2 but less than 2% weight by weight, increase in weight is more than 0.2% weight by weight but less than 2%, then it is known as a slightly hygroscopic. Okay, now if increase in the weight of that compound is more than 0.2% but less than 15%, if increase in the weight of that compound by absorbing moisture is more than 0.2% but less than 15%. ये याद रखो, less than 2%, less than 15%. Okay, so if increase in weight is more than 0.2% weight by weight but less than 15% weight by weight, that compound is known as a hygroscopic. If increase in weight is okay, if increase in weight is more than 15%, if increase in weight is more than 15%, then it is known as a very hygroscopic. Now if increase in the weight is, if it increases the sufficient water to become a solution, then it is known as a deliquescent. So this was question in GPAT 2021. इसके ऊपर क्वेश्चन आ चुके हैं. So remember slightly hygroscopic मतलब क्या है? So there is an increase in weight up to 2% but less than 2%. Okay? So increase in weight by absorption of water is less than 2%, that is a slightly hygroscopic. So if increase in weight of that compound is okay, if increase in the weight of that compound is less than 15%, okay, then it is known as a hygroscopic. If increase in weight of that compound is more than 15%, then it is known as a very hygroscopic. Okay, and if it absorb water sufficient water to become a solution known as a deliquescent. तो ये आपको याद रखना है. Following methods are used to determine. So what are the different methods which are used to determine the moisture content? Moisture content determination के लिए कौन से मेथड है? Following methods are used for moisture content. So basically there are two methods. First one is known as a Curl-Fisher titration. Then your IR analysis is also there. Infrared spectroscopy is also there. Thermogravimetric analysis is also there. So first method, thermogravimetric analysis. Second method, gas chromatography. Third method is nothing but the gravimetric analysis. Fourth method is nothing but the Curl-Fisher reagent. Curl-Fisher reagent. So you have to remember this method. Methods used for measuring the moisture content. कितने मेथड है? चार मेथड है. थर्मोग्राविमेट्रिक एनालिसिस, गैस क्रोमेटोग्राफी, देन ग्रेविमेट्री, ओके, आईआर भी आता है उसमें, आईआर स्पेक्ट्रोस्कोपी और कर्ल फिशर reagent. Out of that curl fisher reagent is very important. They can ask question on this. So what are the composition of curl fisher reagent? इसका composition क्या होता है? So remember, curl fisher reagent is composed of sulfur dioxide, then pyridine, okay, pyridine, sulfur dioxide, iodine, and methanol. Okay, pyridine, sulfur dioxide, then iodine and methanol. So remember these four content of this curl fisher reagent. They can ask you one of the following not a content of curl fisher reagent. Okay, they can ask you a question. अरे भाई लेक्चर में हो, समझता नहीं क्या आपको? अरे लेक्चर में हो ना, आपको जो भी काम है, आपको जो भी काम है 6-7 बजे के बाद फोन कीजिए. Then curl fisher reagent. Now next one is a Ventoff plot. So Ventoff's plot. So Ventoff's plot is nothing but if you plot a molar solubility of compound on y-axis and versus temperature, so you will get this Ventoff's plot. So इसके ऊपर भी क्वेश्चन आता है, what is meant by Ventoff's plot? So plot of temperature versus molar solubility. A molar solubility, temperature at which solubility and vapor pressure of polymorph are same. What is meant by molar solubility? It is nothing but the solubility temperature at which the solubility and vapor pressure of your polymer it is same. Then it is known as a molar solubility. If you plot a temperature versus molar solubility, that you will get a Ventoff's plot. Okay? Now bridging solvent is a solvent which is used to convert one polymorph into another polymorph. ठीक है, एक पॉलीमॉर्फ को दूसरे पॉलीमॉर्फ में कन्वर्ट करने के लिए ब्रिजिंग सॉल्वेंट यूज करते हैं. इसके ऊपर भी नाइपर में ही क्वेश्चन आता है, जीपैट में ही क्वेश्चन आता है, ब्रिजिंग सॉल्वेंट. ठीक है, एग्जांपल्स भी बाद में देखेंगे. नाउ फाइन पार्टिकल कैरेक्टराइजेशन. ठीक है, फाइन पार्टिकल कैरेक्टराइजेशन. नेक्स्ट पॉइंट है फाइन पार्टिकल कैरेक्टराइजेशन. तो इसमें पार्टिकल साइज डिस्ट्रीब्यूशन, डेंसिटी, पोरोसिटी, ओके, देन टैप डेंसिटी, एंगल ऑफ रिपोज, फ्लो प्रॉपर्टीज. सो बल्क फ्लो, then formulation, homogeneity, and surface area. These are the control process like distribution, dissolution, chemical reactivity is affected by size, shape, and morphology of your particle. So bulk flow, formulation, homogeneity, surface area, and control process like dissolution, chemical reactivity. Now these processes can be affected depending on the size, shape, and morphology of your particles. Okay. Now methods which are used for the determination of particle or method which are used for the particle characterization, these are nothing but the first method is nothing but the sieve analysis. So by doing or by subjecting your compound to sieve analysis, you can get the idea regarding the particle size of that molecule. Okay? So sieve analysis is used when particle size more than 1000 micron. Oh sorry, particle size more than 100 micron. अगर आपका particle size 100 है, 100 micron है, उससे ज़्यादा है तो sieve analysis. Now light microscopy is there, or it is known as optical microscopy. Then culture counter method, we have seen this in a micrometric chapter. Okay, anybody want to see, you can go and see. Culture counter method is there. Then this HIAC, it is known as a stream counting method. Now BATE theory means Brunner-Imet-Taylor theory. So it is a theory for adsorption, determination of surface area of particle by adsorption. We have seen in the physical pharmacy. So BATE equation. So remember over here, one nitrogen atom or one nitrogen molecule is equal to, will occupy a area of 16 angstrom unit. Okay, so nitrogen atom, liquid nitrogen at minus 196 degrees, liquid nitrogen at minus 196 degree. It is subjected. Okay, powder is subjected to this liquid nitrogen and it is analyzed. So one nitrogen atom will occupy 16 angstrom unit area. Okay, remember. इसके ऊपर भी क्वेश्चन आ चुके हैं जीपैट में, नाइपर में. सो बेट इक्वेशन मींस Brunner-Imet-Taylor equation, determination of particle surface area by adsorption method. Now second is the same, that is scanning electron microscope. So it is used for the study of surface morphology and texture, कि आपके पार्टिकल का सरफेस कैसा है, स्मूथ है, रफ है, कैसा मार्जिन हुआ है, सब. So that is used for the surface morphology and texture. Now to make, normally what happens, most of the powder or most of the compounds, they are non-conductive, conductive nahi rehte ho. To make that compound conductive, gold plating is done. So compound is gold plated to make a particle superconductive. इसके ऊपर भी क्वेश्चन आता है. Now what do you mean by bulk density? Bulk density means what? Mass upon volume. अगर कोई कंपाउंड का मास 5 ग्राम है, वॉल्यूम 6 है, 5 divided by 6, that will give you density. So bulk density means it is a, okay. So it is again important in case of high dose of drug. किसी ड्रग का डोज ज्यादा है तो उसके लिए बल्क डेंसिटी आपका, you have to check the bulk density of drug if your dose of drug is high. Now apparent bulk density is nothing but the gram per ml. It is given by in the gram per ml, and it is calculated on the 40 mesh pre-sieved powder. मतलब आपका जो पाउडर है वो पाउडर लेना है आपको, उसको 40 sieve से पास करना है, तो उसके बाद में जो नीचे पाउडर आएगा उसका आपको बल्क डेंसिटी कैलकुलेट करना है. मतलब बल्क मतलब weight upon volume, that is bulk density. True density is nothing but the bulk density minus wide volume. Wide volume means interparticular volume. We have seen in the, and tap density means it is determined by pouring the known quantity of powder in a graduated cylinder and there is a tapping of that. Okay? Tapping not less than 100, not less than 1000 tapping. तो ये क्या याद रखो, इसके ऊपर भी क्वेश्चन. Flow properties of powder is determined by compressibility index, angle of repose. ओके, तो फ्लो प्रॉपर्टी कैसे करेंगे, कंप्रेसिबिलिटी इंडेक्स. फ्लो प्रॉपर्टी कैसा है, कंप्रेसिबिलिटी इंडेक्स क्या है उसका. ओके, उसके बाद में हाउसनेस रेशियो क्या है उसका, उसके बाद में एंगल ऑफ रिवर्स कितना है वो पाउडर का. सो डिपेंडिंग ऑन दीज थिंग्स, सो फ्लो प्रॉपर्टीज इज मेजर्ड, इट इज डिटरमाइंड बाय these methods like compressibility index or Car's index, then angle of repose, and then your that Hausner's ratio. So see it. So there are two kind of powder. Either powder may be free flowing. तो powder क्या होगा, free flowing होगा. That means it can easily flow in the from hopper to die cavity of tablet. Now non-free moving powder or non-free flowing powder. So it is known as a cohesive powder. अगर powder non फ्री फ्लोइंग है तो उसको कोहेसिव बोलेंगे. ये पाउडर इज फ्री फ्लोइंग, इट्स ओके. कंप्रेसिबिलिटी और कार्स इंडेक्स, फर्स्ट वन इज कंप्रेसिबिलिटी और कार्स इंडेक्स. इसको कार्स इंडेक्स को ही कंप्रेसिबिलिटी इंडेक्स बोलते हैं. तो कंप्रेसिबिलिटी इंडेक्स और कार्स इंडेक्स, रिमेंबर, it also measures the flow property of the powder. It is a measurement of flow property or it is an indicator of flow property of powder. So what is Car's index? Percent compressibility is equal to, so it is a formula like tap density minus true density divided by tap density into 100. ठीक है, यहाँ पे देखो ये compressibility, percent compressibility is equal to tap density, okay, minus true density divided by tap density. Tap density or my tap density minus bulk density divided by tap density. So here tap density and bulk density. So PT rho T is nothing but the tap density and rho 0 is nothing but the bulk density. So tap density minus bulk density divided by tap density into 100, that is the compressibility index or it is known as the Car's index. ये डिटरमाइन करना पड़ता है आपको पाउडर का. So percent compressibility index. So if powder is having Car's index in between 5 to 12, if powder is having Car's index between 5 to 12, so it is known as the excellent flow. Okay, powder is having excellent flow properties. If Car's index is between 12 to 16, okay, then powder is having good flow properties. If compressibility index is between 18 to 21, so you can say the powder is having passable flow properties. Now if Car's index is between this 23 to 35, then you can say it is a poor flowing powder. If Car's index of powder is between 33 to 38, it is a very poor flow powder. Okay. If Car's index is more than 40, okay, Car's index is more than 40, it is a very poor, very poor flow, or you can say cohesive powder. Okay. So it is a Car's index, it is a measurement of your flow property of powder. Remember formula याद रखो, Car's index is equal to tap density minus bulk density divided by tap density into 100. Then next property or next property to measure the flow of your powder, that is Hausner's ratio. Hausner's ratio ये क्या है, tapped bulk density divided by poured bulk density. So Hausner's ratio means it is a ratio of tap bulk density divided by poured bulk density. So Hausner's ratio will vary from 1.2 to 1.6. So it is 1.2, it is a free flowing. If it is 1.6, it is a cohesive powder. Okay, you can see again here. So Hausner's ratio and type of flow. So if Hausner's ratio is between this 1 to 1.11, if Hausner's ratio is between 1 to 1.11, the flow is excellent. Powder flow is excellent. Then if Hausner's ratio is between this 1.12 to 1.18, you can say powder is a good flowing. Then if Hausner's ratio is between this 1.19 to 1.25, you can say powder has a fair flow. Then Hausner's ratio is if it is between 1.26 to 1.34, is possible. ये सब कुछ याद नहीं रखना है आपको. एटलीस्ट जो एक्सलेंट फ्लो के लिए कितना रिक्वायरमेंट है एंड पुअर फ्लो का कितना रिक्वायरमेंट है उतना भी याद किया आपने तो आपका काम बन जाएगा. एग्जाम में क्वेश्चन आता ही आता है. ठीक है. सो यहाँ पे Hausner's ratio जो जो आप याद नहीं रख सकते ज्यादा, उस एक्सलेंट वाला, गुड वाला एंड पुअर वाला याद रखो. एक्सलेंट वाला, गुड वाला एंड पुअर वाला. यहाँ पे बेस है. तो एक्सलेंट वाला याद रखो, गुड वाला याद रखो एंड पुअर वाला. So if Hausner's ratio is between 1.19 to 1.25, so that flow is nothing but the fair flow. If it is between 1.26 to 1.34, so it is a possible. Then again, if Hausner's ratio is between 1.35 to 1.45, so it is having poor flow. So if it is more than that, is again a very poor flow. Then if it is Hausner's ratio is more than 1.60, it is having very poor flow. Means cohesive powder. Yeah, Hausner's ratio के बारे में. Now next one is nothing but the angle of repose. What is the angle of repose? So angle of repose is nothing but the, इसका एक फॉर्मूला होता है tan theta is equal to 2h divided by d. 2h मतलब क्या हाइट एंड d मतलब क्या डायमीटर. तो वो उसमें आपने you might have performed one experiment in your physical pharmacy lab. So what you have to do, you have to take a 5 gram of powder, you have to pour that powder from funnel below which there is a one paper kept. Okay, and whatever the pile is formed, you have to measure the height of that pile and you have to make a circle around that pile and you have to measure a diameter, and now you have to calculate the angle of repose of that powder. That is nothing but tan theta tan theta is equal to 2h divided by d. So h is nothing but the height of powder pile and d is the diameter. That is known as angle of repose. There are two kind of angle of repose. Static angle of repose, which is measured by fixed funnel method. There are two type of angle of repose. आपको ये नहीं किसने बोला होगा. Static angle of repose is measured by fixed funnel method. Panel नहीं है यहाँ पे, funnel है. Fixed funnel method and fixed cone method. So static angle of repose is measured by fixed funnel method and fixed cone method, and then kinetic or dynamic angle of repose is measured by rotating cylinder method and tilting box method. So remember this method. इसके ऊपर क्वेश्चन आ सकता है क्योंकि ये थोड़ा सा नया पार्ट. Okay. Now we'll see the angle of repose and its plot. So if angle of repose of powder is between 25 to 30, अगर आपके powder का angle of repose इसमें आता है 25 to 30, so you can say the powder is excellently. So if powder is having angle of repose in between 31 to 35, so it is a good flow. Okay, powder is having 36 to 40 angle of repose, that means powder is having fair flow. If angle of repose is between 41 to 45, that means powder is having possible type of flow. Then if angle of repose is between this 46 to 55, then it is a poor flowing powder. If again if angle of repose is between this angle of repose is between 56 to 65, that means powder is very poor flow. Powder is having very poor flow. If angle of repose of your powder is more than 66, so it is very poor flow. यहाँ पे ये क्या रखो, एंगल ऑफ रिपोज लेस देन 30. आप ये वैल्यू छोड़ो, आप अपने हिसाब से बनाओ. लेस देन 30 एक्सलेंट, लेस देन 30 एक्सलेंट. ओके, 30 to 40 you can say 30 to 30 above you can say good. Okay, ऐसा 35 to 40 fair, then 40 to 45 possible, then 45 to 55 poor, then 55 to 65 very poor, and more than 60 is more than 65 very poor. ऐसा थोड़ा सा ट्रिक बना के रखो यार, because you have to remember they can ask direct question, बहुत बार पूछा है. Next one is a particle size determination method. We have already seen this method in the micrometrics. Okay, so there are different methods for measurement of particle size. First one is sieving method. So in this case you use O1 wires. ठीक है, आपका जो सी होता है, आपका जो सी होता है वो क्या होता है, O1 wires का होता है. So you can analyze particle size in a range of 20 to 20 to 1,25,000 micrometer. Then again by sieving method. Now in this case sieve are electro etch, मतलब यहाँ पे क्या है कि इलेक्ट्रिक बीम जो है फोटो इच का मतलब इलेक्ट्रो इच या फोटो इच, मतलब लेजर बीम ये एक पर्टिकुलर क्लॉथ पे ऊपर डालते हैं और लेजर बीम के थ्रू उसके ऊपर परफोरेशन बनाते हैं, छेद बनाते हैं. सो दैट इज नॉट द इलेक्ट्रो इच और फोटो इच. Sieving, electroetched or photoetched, you can determine the particle size in range of 5 to 120 micron. Now, sieving again perforated plate sieve, so you can see you can measure the particle size in range of 1000 to 1.25,000, 1,25,000. By using optical microscopy, you can analyze the particle size in the range of zero point 0.5 to 150 micron. Now, by using electron microscopy, you can analyze the particle size in the range of 0.001 to 5 micron. By using sedimentation gravity method, you can analyze particle size in the range of 1 to 50 micron. Or you can also by centrifugal sedimentation method, you can analyze particle size in the range of 0.01 to 5 micron. Then for culture counter method, by using culture counter method, the particle size analysis is in a range of 1 to 200 micron. By using this light scattering, you can analyze 1 to 1000 माइक्रोन पार्टिकल साइज रेंज. कभी कभी इसके ऊपर क्वेश्चन आता है, ये ऑलरेडी अपन ने फिजिकल फार्मेसी में देखा है. सो दैट्स व्हाई आई एम जस्ट देयर आर these are the examples of compressibility and flowability of certain pharmaceutical additives. ये आपने जहाँ पे फॉर्मुलेशन में ये यूज करते हैं मटेरियल, जैसे सेल्यूटेप, ये स्टार एक्स लैक्टोज मोनोहाइड्रेट वगैरह. So they are used for a certain purpose, for example diazo diluent or directly compressible diluent or disintegrant or binding agent, etc., glidant, okay, opacifying agents. Examples of certain examples of material and their compressibility and flowability. Celluloid J, it has a 11% compressibility and it has an excellent flowability. Again, this N compress is also having excellent flowability. Star X 1500. It is a directly compressible starch. Then lactose monohydrate. So it has a 19% compressibility and it is a fair and possible flowability. Then maize starch and dicalcium phosphate, which dicalcium phosphate of coarse grade. So it will have compressibility in between 26 to 27 and it is a poor flowability. Titanium oxide is used as a opacifying agent. Then magnesium stearate is a lubricant, it is a glidant and it is having compressibility of 36%. It is having very poor flow. Titanium dioxide is having also 34% compressibility, poor flowability. Okay. Dicalcium phosphate, very poor flowability. Talc may be very poor flowability. Now next part is I will show the